So it's obviously a great pleasure to be here. And what I'm going to do is to update you a little bit on our efforts in trying to understand the developmental and neurobiological basis of individuality. And you heard a lot about individual variability and its potential effects in a genetic sense from Daniel, who actually said a lot and repeated during his talk multiple times the idea that not all the variation between individuals in terms of their risk for developing certain neurological or neuropsychiatric disorders was actually genetic. And so where does that non-genetic component of variation come from? Does it matter to individual behavior, to personality traits? Does it matter to disease risk, and so on and so forth? And so this is one of our attempt basically to address this question using the fruit fly as a model system. And so let me start with the sort of the basic idea. And the basic idea is we're all different within any given species. All individuals are different. And when we talk about individual differences, we talk about consistent differences. That is to say that a trait that is yours over time, that is stable in you, but that is different between you and everyone else. And that is a trait that you could attribute as a personality trait to you. And we know that we are, of course, genetically different as individuals. You heard from Dan Gashwin this morning a lot about this and how these genetic differences now we understand in fairly obvious ways can contribute to making us different from one another. We obviously have experiential differences, and those experiential differences, whether they are short or frequently very long-term plasticity-dependent events that change the way our brain works, that change the way our circuits are wired in a functional sense, and that creates differences in our behavior that would be attributed to us as a personality trait. There's actually a third source of individual variability. And to developmental biologists, this is, in fact, almost trivial. In neuroscience, it hasn't really caught up very much. And that is, in fact, what developmental biologists call stochastic developmental noise. Every biologist knows that biology is intrinsically noisy and unavoidably so. And in fact, that noise in developmental processes at every level, molecular, gene expression is noisy, assembly of enzyme complexes is noisy, and so on and so forth. And this noise at different levels, interactions between cells that are noisy, and that noise at different levels results in essentially stochastic differences in developmental processes that cannot be a priori predicted by genomic sequence alone. And the idea was that perhaps these intrinsic developmental variations that are uncontrollable and unpredictable play a role in wiring the brain during development such that two or more genetically identical individuals would develop slightly different neural circuits, and that those differences in neural circuit development in those individuals would result then in functionally different individuals that are stably different from one another because the brain has been wired slightly differently. And so that's the basic idea we wanted to test. If you start from genetically identical brain, because of developmental molecular noise, you would end up with slightly different wiring diagrams for exactly the same circuit. And if that circuit is involved in controlling an innate behavior, you would predict that you can then correlate and eventually try to establish some causal link. And we heard from Marcus this morning that correlation and causality are not always going in one or the other direction. But anyway, try to establish some sort of a causal link between the wiring diagram and the behavior of the animal. So that's the challenge, right? So the first challenge is, how do you find a neural circuit that you could look at in hundreds of individuals or more in an organism where you can actually generate organisms that are genetically identical? And we were lucky because actually just in our work on neural development, we had chosen a neural circuit in the fly brain, which at that time we had no idea what it was doing functionally. It just looked very pretty, and that to me was reason enough to work on it. And it turns out that the circuit displays intrinsic variability between different individuals produced by the mating of one male and one female. So ostensibly genetically identical siblings. And this was creatively named the dorsal cluster neurons because they're dorsal and they're clustered. And it turns out that the number of cells varies not only between individuals, but actually between the left and the right side of the same brain. So almost by definition, a non-genetic variability. So not only is the number of cells variable within a range, of course, but actually these cells turn out to do contralateral innervation of the visual system of the fly. And they make a choice. They're axons. One axon per cell crosses the brain and decides to innervate either an area called the medulla or an area called the lobula. And independent, and we can show this statistically, independently of the variation in the number of cells, there is then variability in the number of axons that decide to innervate one target and the other, or the other. And because the variability is is independent on the left and right side of the brain, this automatically results in asymmetry in every aspect of the circuit. And then asymmetry in the number of cells, asymmetry in the number of axons that target the medulla, asymmetry in the number of axons that target the lobula. So what we tried to understand at first was where was this sort of variability coming from during development. And it turned out that what these neurons are doing are essentially trying to compete for innervating the medulla. So early in development, all the axons that come from one cluster on one side arrive in the medulla. And then they engage in a competitive interaction using notch signaling, which is an intrinsically stochastic process by which a number of cells decide to adopt different fates in an unpredictable fashion. The only thing you can predict about notch activity or lateral inhibition is that one cell will adopt one fate and all the other cells surrounding it will adopt the opposite fate. But which cell will adopt which fate is unpredictable. And so it turns out that every few of these DCN neurons are doing this. And out of four, 5, or 6 cells, one axon would remain in the medulla, and all the rest would go back to the lobula. So this intrinsically stochastic process results in a highly patterned neural circuit, but which is, by definition, variable between individuals. So the next challenge was to try to understand, so this and a series of other, a couple of other papers led us to conclude that molecular noise and variation are used by developmental rules. So what BrainWire really cares about is following a set of developmental algorithms in order to establish things that are robustly active. They don't have to be precisely the same every time. And that actually, if you think about it from an evolutionary perspective, makes a lot of sense. But this actually underlies then the inevitable generation of variation in neural circuit formation. And that these rules, therefore, if we would understand the molecular underpinnings of these rules, we could use these molecules to generate different circuit patterns and ask about their implication for behavior. So the next problem then is, can we find behaviors in flies, innate behaviors in flies, that we could describe as showing personality traits? That is to say that a response of an innate behavior in a given fly is stable throughout the life of that fly, but is different between one fly and the other. And if so, can we actually use this intrinsically variable circuit during development to query the relationship between neural circuit variability and behavioral variability? Okay, so for reasons I won't go into, and after trying a lot of things, we discovered that using this very, very simple assay in which a fly is placed in the middle of an arena that it cannot get out of, and is looking at a black stripe on a white background, on 2 stripes actually, 180 degrees away from each other, that fly spends its time, as you can see here, walking back and forth between those 2 stripes. It's a very simple innate response to basically dark edges on a white background. And what you can do, what Gerrit Linneweber, who then developed this assay in the lab, what you can do is you can, of course, track the behavior of a single fly. You can track the behavior of this single fly over many times. You can take the fly out and test it tomorrow, the day after, 3 weeks later, the same fly, and ask what does the behavior of this fly look like. You can draw a heat map of the multiple repeats of these, and you can see that the behavior of one fly is fairly stable. And more importantly, and so then you can, sorry, before I get to that, you can then ask what are the parameters that describe this behavior. And of course, you can extract from tracking a fly or any animal hundreds of different parameters. But the question is, what is the minimal number of parameters that you need in order to describe the stability of the behavior of this one individual? And it turns out that we can actually narrow everything down to about 8 different parameters. You don't have to worry about what they are. And 7 of those, except the distance traveled, 7 of those are completely stimulus dependent. So if you remove the stripes, the flies stop. You cannot predict what the fly will do from one trial to the other. The distance walked, how much a fly likes to walk, is an intrinsic property of the fly. It's completely stable, but does not depend on the presence of a stimulus. So it's a very nice control parameter for us to look at, because it should be independent, essentially, of the wiring of that particular circuit that controls the rest of the parameters. So we have the parameters, we have the behavior, and we can now begin to ask, is it stable in one individual? Does it vary between individuals? And the answer is absolutely yes. So what you see here is a heat map, which is an average of 3 different days of tests, and we've done this up to 3 weeks, and the results are the same. We get correlation coefficients that range between 0.6 and 0.8 for any given parameter in any given fly. And so you see here, we classify those flies as having strong responses to the stripes, or weak responses to the stripes, sorry. And that's kind of an arbitrary division. It doesn't matter. And whether the flies have what we describe as strong responses, that is to say they walk back and forth between the stripes, or weak responses where they meander a lot or actually go to the left side where the stripes are here, or sort of walk all over the place, it doesn't matter. Whatever their behavior is, it's stable over time. Okay? So we have now a simple innate reflex that these flies have, but the method or the sort of pattern of following that reflex is completely individual to that particular fly, and different between different individuals. This is what we were looking for. And the question is, how independent of genetic background is this individual behavior? Now, what I showed you were data from an inbred strain of flies that we keep in the lab, but inbred in the sense that it's the same strain that's breeding to itself over many, many generations in the lab, et cetera. So not inbred in the sense that it actually was collected in the wild from one animal or one pair of animals and then generated. So we wanted to ask whether individual variability would persist in the face of a truly inbred population that is in the wild, so it has not been selected in the lab. And second, which was the really challenging experiment, in the face of an isogenic inbred population. And so this very simple slide summarizes enormous amounts of work by Garrett in order to so he's tested about 4 or 5 different inbred populations that we have in Drosophila that were collected somewhere in North Carolina and then established as inbred line initially for the purpose of SNP sequencing. And you can see that when you plot the average population behavior between these 2 lines, it's completely different from one another. So these flies have, on average, as a population, have a very strong sort of centralized stripe response. And these flies kind of like to walk around the edges. And yet, within that population, which is inbred, and this one, which is isogenic, despite the differences in population behavior, which are almost certainly due to genetic factors, the stable behavior of individuals and the differences in that behavior between individuals persist. So even when you make the population isogenic, you cannot wipe out stable individual differences in behavior. So the question now is, to what extent does neural circuit variation underlie behavioral variation? Obviously, I presented this behavior to you because we later found out, as I'll show you in a second, that circuit that we started out looking at in terms of trying to understand the neurodevelopmental basis of neural circuit variation turns out to actually regulate this particular behavior. And so we can now study the correlation. And so the experiment is, quote unquote, very simple. You take one fly. You take hundreds of these 1 flies. You test it over 3 days. So on day 1, you put the fly in the arena for 15 minutes, you take it out, you put it on a vial on its own, you come the next day, you do the same experiment, the third day you do the same experiment, you establish a pattern of behavior for that one fly. Once you've established a pattern of behavior for that one fly, you thank it very much, you sacrifice it, and you look at its brain. And this actually been working quite a bit on trying to automate the description of the anatomy of the neural circuit. And we care about 3 things. We care about cell numbers on the left and the right side of the same brain. We care about the number of, or the degree of innervation of that distal target area called the medulla, and the degree of innervation of that proximal target area called the lobula. And we care about the asymmetry in this innervation pattern. And then we ask, to what extent does the anatomy correlate with the behavior? And what aspects of this anatomy, particularly the asymmetry in this anatomy, can predict the behavior? And so this is a table that summarizes some of the data. We're still doing quite a bit of quantification and so on, but this suffices to show you this. Basically, the bottom part is grayed out because it's simply the sort of other hand of the top part. So, for example, we talk about medulla. Where is this? Medulla left over right, and this is right over left. So it's the mirror image of the same data. So if you have a strong correlation here, you would expect a strong anti-correlation here, and that's what you see. The bottom line of this data is that, of course, not of course, but we're very happy to see that there are very strong correlations between specific anatomical parameters in a given individual and the way that individual behaves. So this is to say that the anatomy of the circuit predicts the behavior of the individual. And it breaks down in rather interesting ways. So what I've grouped in red here are parameters that seem to show either strong correlation or strong anti-correlation with particular, so anatomical parameters with particular behavior parameters. And the interesting thing is, when you look at things to do with the medulla, with the asymmetry of the innervation of the medulla, you recover aspects of the behavior, parameters of the behavior that have to do with motion. So what is the fly doing during motion? How is it oriented towards the stripe during motion? Okay? How far from the center, the idealized center, is it during motion? And it turns out that the things that I'm showing you in green here, where either the asymmetry in cell number or the asymmetry in the innervation of the lobula, which are not independent from one another for reasons I can explain later, turn out to predict where the fly is relative to the stimulus while it's static. So different aspects of the circuit control different elements of the behavior, whether the fly is actually, quote unquote, making a decision where to go. So it's looking at the stripe of interest but without moving, versus when it begins, it actually initiates, executes the decision to move towards that stripe in a specific pattern at a specific angle. And the key parameters, as is summarized here, so all significant behavior anatomy correlations, again, so the wiring of an individual predicts the behavior of that individual, and the key element is the asymmetry in that wiring pattern. Medulla innervation asymmetry correlates with behavioral preferences in motion, lobula innervation asymmetry during pauses. So this is telling us that DCN anatomy correlates or predicts the behavior of an individual. Are the DCNs actually required for the behavior? And the answer is yes. So if you silence only the aspects that innervate the medulla, the population behavior will change, and most of the flies, the vast majority of the flies now, become completely centered in their approach to the stripe. If you silence the activity of all the cells, you get exactly the opposite phenotype. But because majority of the cells innervate the lobula, that makes sense, right? So this tells us that these neurons are involved in this behavior, and that the way you manipulate the activity of these neurons changes the way the animals behave. But that doesn't tell us that if you manipulated the anatomy of the circuit, that changes the way the animals behave. And this is that experiment here, in which, because we knew that the decision to innervate lobula versus medulla is dependent on the notch receptor, we can now actually inactivate the Notch receptor in all or most of these cells, and therefore prevent the axons that early on innervate the medulla from now going back. So you create an animal in which the vast majority of the axons are in the medulla. And this creates a change in the behavior that mimics what you do when you inactivate the circuit. Okay? When you inactivate most of the cells, that is to say, when you inactivate lobular innervation. So either by doing that electrically or by doing that developmentally, which you rewire the circuit, you can rewire the behavior, suggesting a very strong causal relationship between the wiring pattern and the behavior. And we're in the process, of course, of analyzing this at single fly resolution, so we can show that even though you can change the average behavior of the population, you would still be able to predict the behavior of one individual from the wiring pattern of that one individual. So the message is very simple. Stochastically generated neural circuit asymmetries in individuals that are genetically identical creates individual variability in behavior. And I would therefore extrapolate from that to say that it is not unlikely that the same types of changes in the development of the human brain in people that may be equally genetically disposed to one thing or the other, be it anywhere on the sort of neurodiversity continuum, might then explain why those individuals, despite similar genetic predispositions, end up developing or not different types of behavioral responses. So I want to thank the people who funded. We've recently moved from Leuven to Paris, and so past and present funders, especially, of course, the Paul G. Allen Frontiers group for believing in the possibility of this project and funding it. I actually live 500 meters to the left of here. I love Paris. And this is the ICM where we work now, and I just want to pay homage to the fly and its 6 Nobel Prizes and counting, having spoken right after a potential future Nobel laureate. And thank you very much for your attention.
We have time for one question.
Bassem, one prediction maybe that comes out of your beautiful work is that there might be some behaviors and thus some circuits that evolution might have selected to be more resistant to the stochastic noise and some to sort of enable flexibility in behavioral space. Do you have any evidence for that? Are there some circuits that drive core behaviors?
Yeah.
Or, yeah.
I mean, there's certainly, evidence for the idea that primary sensory circuits, so the first synapses, are a lot less variable than higher order circuits, so that, olfactory wiring and so on. And that, I think, makes a lot of sense. You found a winning formula. You don't want to. And what's interesting, and I think Daniel also alluded to that, is that a lot of the inter-individual variability in brain anatomy when you do, in humans, when you do MRIs, seems to have to do with higher order neural circuit function. We see that reflected in the fly. I mean, the DCNs are just about the highest order visual circuit that anyone has found so far in terms of where they are in the sort of synaptic number of synaptic stations. And it's certainly the most variable that we've seen so far. Compared, for instance, the photoreceptor neurons when they arrive in the lamina, which make completely unalterable circuit patterns. And so retinal axons arriving in the first optic station is absolutely always 6 photoreceptors per laminar cartridge, et cetera, et cetera. Even the number of synapses they make is highly constrained. So you're absolutely right about that.
Okay, thank you very much.
Yeah.